All right, everyone, we're going to go ahead and get started. Welcome to the final round of the 3-minute thesis competition. We are excited to have you here. I'm Stephen Roth, Associate Provost Dean of the Graduate School. It is my pleasure to welcome you all to this event. I'm excited to see our finalists because as a premier land-grant institution, one of our charges is to actually do awesome and amazing work and tell the public about it. And you know what we don't do so well at R1 campuses? Tell the public about it, right? Or we tell them and we immediately get into jargon and we've lost them like that. So these folks have already won college level competitions, right? So they're already showing their commitment to public discourse, public communication, and so we get to just see all of their amazingness. This is a supportive group. I've already told the presenters, you're here to support excellent scientists and scholars doing their work, and we just want to get excited with them. So these students, they're honing their presentation skills, they're leaning into this commitment for how to best present their work to an educated but non-specialist audience. That's the core of what the 3-minute thesis is. For those of you who've never seen one, hold on tight. They go fast, they're amazing, and you'll see what students can do with just a single static slide and 3 minutes. So, great work that you've already accomplished. You're already winners, but we're excited to see what else you can do. I want to give a special shout out to Robin Kotzker, Masuka Stratton, and the other grad school staff who have helped put on this event. We wouldn't be here without you. I want to give a special shout out to our 4 judges as well, who will be judging the competition today. Appreciate your engagement. We have a past 3MT winner from last year as part of the judging, so we know she knows what she's looking for. So really appreciate that. Now I would like to introduce our MC for the event, Marcia Gunsler-Stevens, Director of the Stamp Student Union, and I look forward to hearing some great presentations. Thank you all.
Perfect. What a joy to do this again. I have to tell you, I think this is my 10th time. So I even know the past winner and know something of her research because I told everyone about it after I heard it. So I will be repeating much of what I learned today to anybody who'll listen to me, including a young woman I met yesterday sitting on a park bench on Hornbeck Plaza that I said, Alex, you need to come today. This is the most amazing program. And your lunch looks really good. So my name is Marcia. And I will tell you that this truly is an extraordinary experience, and you will make it such. So take a deep breath, all the presenters, because we are going to learn so much from you, and I will be so grateful for that. Let me tell you what our schedule is so that anybody who's setting their watches. We start with 11 presentations, right? They're master's pre-candidacy students, right? Then there's a halftime, no band, just ice cream. And so we'll take a little bit of a break, about 1:45, and just the people who've already presented will be like, whoo, I can have some ice cream. The ones who haven't yet will be like, I can't have any ice cream. So, and then at 2 o'clock, we'll start those last 10 presentations, which are post-candidate folks. And what I really love is that we're live streaming this. And I don't know, is anybody's family member or friend watching while you are live stream? Absolutely, you gotta do that. And for the people in the room, there are hard copies on several chairs of the program run of show and who will be going when. But we'll always introduce the presenter before they start and tell you where they are. And then there is a 3-minute thesis, 3MT website. You'll want to remember that because that's where the winners get announced in the not so distant future. There's no winning combo today. You won't know. So 3MT, but you can also pick up the live stream there, and you can also pick up the recording later so you can show your family and friends. So as Steve said, already amazing winners here. But some of the most amazing winners are in the back of the room, which are the judges. So you know who you're talking to. I want to introduce all 4 of them. Jinglei Liang, and just give a wave. Beautiful. Is a PhD from 95 Geographical Sciences founder of the Earth Resources Technology, a company with more than 30 years experience offering space and earth science, enterprise and engineering and mission operation solutions, and is a member of the University of Maryland Foundation's Board of Trustees, which is so fabulous. Ray Phaneuf is a professor in the Department of Materials Science and Engineering, a regular judge, and holds an affiliate professor position in physics and ECE. Earned his PhD from the University of Wisconsin-Madison, a good Midwestern place. Among his most notable grad students, and whose research I do remember, Dr. Amy Marquardt, who was UMD's first 3MT winner and went on to win the international competition. No pressure. You could be the repeat. This is a former and current judge, so we're so grateful. Emmy Warner is right there. Emmy received her PhD in Environmental Health Sciences at the University of Maryland in Public Health and the Department of Global Environmental and Occupational Health just in 2025, so just a minute ago. And she was one of the 2025 3MT Award winners. Her talk, Harvested Rainwater, tapping into an alternative irrigation water source, was so darn intriguing, and boy, do we need that information now. A postdoctoral researcher in the Department of Plant Science and Landscape Architecture is what this amazing person does. And Dennis Van Engelstorp, Dennis, right there, is a professor and member of the Department of Entomology, Honeybee Lab. Among the lab's most notable grad students that I learned so much about honeybees when she did. Dr. Sammy Ramsey won the International 3-Minute Thesis Competition in 2017 and the judge's first place prize and People's Choice Award. Big sweep. And so this is a person with a real investment in it because he knows a winner when he's seen it. So we're really grateful to these judges. After each person, we'll vamp for like a minute or two just to give them a chance to dump their thoughts on computer, and then we'll go with the next person, but that'll give the person that just finished a chance to take a deep breath, and the person coming up to know, ooh, I should take a deep breath. So thank you for being here. One quick thing, just do me a quick favor. This is just a little data collection. How many of you are grad students yourselves? Look at that, you're among friends. How many of you, that's right, how many of you are faculty members? Proud faculty members, probably of some of these students. How many of you are family members who came because they let you come? Woohoo! And how many of you don't know how you got here, but you're so glad you did? Nicely done. So, the judging panel is going to select 6 winners, and the announcements, again, will be made on that 3MT website, just to make you think about this. They're going to receive $1,000 plus professional production of their 3MT speech so that can go into the competition that follows. That announcement is on this Friday. You'll have your taxes done. You can look it up. Rules of the competition, and you heard Steve say this, 1 slide, right? 1 static slide. That is an international rule, and it started in 2008, so you don't mess with something that old. And then the other rule, no poems, no raps, no props, no costumes. Other than that, 3 minutes to teach us amazing things. And it originates because the very good people from Australia and Queensland were suffering a severe drought, and they decided that to conserve water, you could do a 3-minute shower. And so why not measure 3 minutes by all the information you could dump in your shower? I'll tell you about honeybees. So we are so grateful to have our first group of people. Charlie Fisher is in electrical and computer engineering. And Charlie Fisher, I'll give you that, Will is pre-candidacy, and we'll be talking about, hear me out, you can train your brain.
Can you hear me? Okay, great. Just checking. Imagine looking through a camera. At first, the image might be a little blurry, but as you move the lens, you can decide what to focus on. Maybe it's the flower or the fine details in the background. I like using this visual analogy to explain how we listen in noisy environments, because what's happening with our eyes is the same thing that's happening with our ears. Right now, your brain is moving the lens to pick up on my voice and blurring out all the background noise. If you're young, this is pretty easy. But as you get older, it becomes more difficult. And it's not just because of hearing loss. As you age, so does your brain, and that makes it harder to focus. This issue impacts a large portion of the older population, like our parents and our grandparents. In order to solve this issue, we need to know what the brain is doing and how we can improve it. And that's what my research focuses on. My project uses something called auditory cognitive training, which you can think of like exercise, but for your brain. It trains your brain to listen in noisy environments, and past behavioral evidence is really promising. But I wanted to understand how the training helps us focus better. What is our brain doing differently? To answer this question, we're taking older adults and recording their brains before and after training. We're looking at how their brains respond to speech with different background noise. We do this by creating a map of the brain response to see where and when the activity occurs. The map tells us how fast or slow the response is, or whether it encountered an obstacle. And by comparing the maps of before training and after training, we can see where the path gets easier. And by doing this, we're essentially showing that the training works by showing how it works. And this opens up new avenues to create better training programs, or maybe even smart hearing aids. But most importantly, we're showing that we can improve the quality of life of so many people. Because imagine what it would be like to wake up one day and you've lost the ability to hear, but only in certain situations. It would become extremely frustrating and may even cause you to avoid those situations altogether. This is what some older adults deal with every day. And while the purpose of this research is to help people hear better, the goal is to bring our loved ones back into the conversation, no matter the environment, to spend a little more time talking, a little more time laughing, and a little more time understanding. Thank you.
Charlie starts us off today with the topic that we were talking about, because after 44 years at working at the University of Maryland, I'm on the precipice of graduating finally. And I said teaching classes when students wore masks was really hard. So I have great hope that Charlie will do research that'll aid me individually, which is so fabulous. Okay, one done, and amazingly so. We're up for our second presenter today. Bridget Moran is a really cool person from the College of Education in school psychology. And the topic today of Bridget's presentation is Conversation Through Stories, Strengthening Parent-Child Dialogue About Screen Use. Ooh, take it away.
In a recent parent email thread, one parent wrote, "Not a day goes by where we don't have a discussion about screen use in our house." This parent is not alone. Many parents report that technology is making parenting particularly challenging today. Parents have tried tactics like setting limits and monitoring use, but screen use is more complex than simply restricting access, because screens also serve important purposes, like facilitating social connection and supporting learning. That means that screen use is not a single problem with a single solution. It depends on the context of use. Is a child alone or with friends or family? The nature of use. Are they passively scrolling or actively creating? And the impact. How does it affect their sleep, mental health, and school engagement? Limits may help regulate behavior, but they don't address motivation or build insight. That's where my research comes in. Storybooks offer a powerful way to open these conversations. When children reflect on characters' thoughts, feelings, actions, and consequences, they can reason safely without feeling judged. My research explored existing children's books about screen use to analyze whether they could serve as powerful tools to facilitate parent-child dialogue. I found that most stories lack the nuance needed to create meaningful conversation. Most stories present overly simplistic messages, focus mainly on the negatives of screen use, and rarely explore characters' inner experiences. I concluded that this, however, could hold its advantages, offering parents the opportunity to fill this gap. Using psychological theory, I made suggestions for how parents can tailor these stories to their child's unique needs and support foundational social-emotional skills like perspective-taking, emotional insight, decision-making, and self-regulation. Most importantly, these conversations can strengthen the parent-child relationship through facilitating connection, communication, and shared understanding. In a world where digital media is inevitable, elimination isn't realistic. But when we combine thoughtful screen limits with meaningful conversations, we move from simply regulating behavior to raising reflective, resilient children who use technology with intention.
How many of you are thinking about stories from childhood at that very moment? Yeah, I was going down that road. Thank you so much, Bridget. Our next presenter, Nayeeb Rashid, is a wonderful person who I will tell on you that you and your wife are both here as doctoral students in electrical and computer engineering. And today, Nayeeb is going to talk to us about the hidden signal in your video call. Take it away, Nayeeb. Good waiting.
How many of you have been in a Zoom call in this past week? Have you ever wondered where the other participants of your call are actually located in? Sometimes maybe you know the answer to that. Sometimes you don't care. And sometimes even asking that can seem a bit nosy. But imagine you're taking a professional licensing exam, or you're talking to a lawyer, who for legal reasons can only consult you if you're inside a particular territory. In cases like this, knowing the location of your video call participants isn't just a mere curiosity, it becomes a security question. Current platforms like Zoom only track your IP addresses, which can be easily masked using commercial VPNs. So we need a more reliable and robust way of knowing locations inside the video calling setup, and that's where my research comes in. For this purpose, we use a power signature called electrical network frequency, which is created by the massive electric grid turbines that are spinning at 60 Hz here in the USA and 50 Hz in other parts of the world. As the energy demand shifts in our electric grid, this signal, it fluctuates slightly around this nominal value, which creates a unique power signature for every region. Now, what makes this signal truly interesting for our use case is that it is automatically embedded into your media recordings. Today's audio and videos capturing devices inherently pick up this signal from the surrounding environmental interference. But so now, since Zoom calls give us both audio and video signals, they present this perfect platform for using a technology like this. But it's not that simple. Think about all the different choices of devices, microphones, the room environments that exist in a Zoom call, all of which impacts this weak, hidden signal. Our research takes all of this into consideration, uses signal processing and AI to securely, robustly, and accurately extract this signal. Now, once we have this signal, it can act as a location tracker. Just from the base frequency itself, we can tell whether you're in the US or in some other parts of the world. Even inside the US, different power grids have different fluctuation patterns, which can help us narrow down your location. And if we want to pinpoint your location even further, we can place sensors at your location and compare the signals. Now, all of this might sound a bit scary, how we are encroaching on your privacy, but while this is a powerful tool, it is only intended to be used in very high security cases where the user is both willing to provide the location and prove the location. By creating a system like this, which can verify your location from something as simple as a video call, we're trying to create a new standard of trust for our most sensitive digital conversations. Thank you.
Thank you so much, Nayeeb. We are so lucky to think about all those Zoom calls we've been on, and everybody knows exactly where you are. You're not really at home, you're in a different place. Our next person, Rayal Smith, comes to us by way of the Department of Chemistry and Biochemistry. And I need to really pronounce this well, or pronounce it, depending on where you put the hyphens. Don't be atmospheric. What really is in the air you breathe. It is a great joy to have you with us, Rayal.
Take a deep breath. Now, what did you just inhale? Most of us would say oxygen, maybe pollution. But here's the truth. Every breath you take contains tiny particles filled with chemistry. Chemistry that scientists still don't understand. Hidden inside these particles is a molecule called oxalic acid. Now, oxalic acid is everywhere. It's in the clouds, it's in airborne particles, it's even in the air that we're breathing right now. These particles are so tiny that they can travel deep into your lungs and can affect respiratory health and even worsen conditions like asthma. But here's the problem. We can measure what's in the air, but we don't know exactly how it got there. And if we can't understand how oxalic acid is being formed, then we can't fully predict air quality or its impact on our health. You see, the atmosphere isn't just empty space, it's a chemical reactor. Inside tiny water droplets, like the ones found in clouds or humid air, pollutants are constantly reacting, and some of these reactions can create oxalic acid. Now, these changes can determine the longevity of the particles in the air, how toxic they can become, as well as its impact on our bodies. So you see, this isn't just chemistry, this is also public health. So, how do we figure this out? Well, my research looks into the real measurements of the air that we breathe. I analyze when oxalic acid appears and under which conditions it's being formed, such as humidity and pollution. And what I found is that the more water that's present in the atmosphere, the more oxalic acid tends to form. And this shows us that these tiny water droplets play a large role in driving these chemical reactions. And this is important because the more that these particles tend to disperse in the air, the more that these oxalic acid molecules tend to be present. So the next time you take a breath, remember, you're not just inhaling air. You're inhaling the result of millions of chemical reactions that are happening all around you. And the better we understand how oxalic acid tends to form, makes us one step closer to making the air around us cleaner, safer, and healthier for everyone. Thank you.
Rayal, I don't know if anybody else in the room is wrestling with the pollen. I was thinking, you're not just talking about pollutants, you're talking about pollen, which was so beautifully done. Our next presenter is Adriana Hammond. Adriana comes to us from the School of Architecture, and her topic is war wounds.
What does it mean to rebuild a place that has been wounded by war? Since 2022, Ukraine has been fighting not only for its territory, but for global recognition of its independent status within Eastern Europe. When missiles strike cities and neighborhoods, they do more than just damage buildings. They rupture memory, ritual, and the spaces where everyday life unfolds. As of this year, UNESCO has reported more than 500 culturally significant sites within Ukraine that have been damaged or destroyed by Russian airstrikes. And with each damaged building is the loss of lived experience. A school with the voices of children, a church where generations of couples were married, and even a cemetery where people go to remember their loved ones. My thesis asks how historic preservation should engage with and respond to this kind of destruction, not just historically, not just technically, but experientially. And to ground this research, I traveled to the historic city of Lviv in Western Ukraine and selected a war-damaged building as a site of study. There, I conducted fieldwork within a phenomenological framework of note-taking and drawing to explore how reconstruction can move beyond strict material fidelity and instead respond to the lived experience. Phenomenology, a philosophical tradition that emerged in the early 20th century, centers the embodied experience. It seeks to describe how we encounter the world through our senses, how meaning arises through touch, sound, memory, and shared presence. While modern architecture famously insists that form follow function, phenomenology instead asks questions. What is the essence of a place, and how does it shape our sense of being? And how can a place foster what phenomenologists name intersubjectivity, or the solidarity between peoples? This thesis proposes that phenomenology offers critical guidance to architectural preservationists in post-conflict environments. Traditional preservation frameworks often define significance through materiality, architectural style, or a notable person, think Thomas Jefferson at Monticello. But buildings damaged by war reveal that significance is not fixed. It evolves. It accumulates meaning through trauma, resilience, and collective memory. Rather than ask how we can rebuild a building in the exact same way, this thesis explores how architecture holds memory, fosters solidarity, and can remain open in an uncertain future.
Thank you so much, Adriana. All right, halftime in the first, shall we just say the end of the first quarter? Here's what I need you to do. I need you to turn to the person in front of you or next to you and introduce yourself. And if you already know the person next to you, you need to tap the person in front of you. And I want you to introduce yourself so that you know who else is here besides you. Go. You only have 2 minutes, so make it a good conversation. You and your new best friend need to come back to order. But hopefully, when it's ice cream time, you could be like, oh my gosh, we're from the same part of the world, or whatever, and you can get ice cream during halftime. Thank you so much for giving the judges a minute, because I was not vamping long enough, and oh my gosh, there's so much to take in. So, thank you so much. Our next presenter is Elizabeth Huang. And Elizabeth is from Geological, Environmental, and Planetary Sciences. I think about what we've all been experiencing watching the dark side of the moon the last couple of weeks almost. And Elizabeth's topic today is a tapestry of stone. Elizabeth.
Tapestries have been used to tell stories for centuries. They're a way by which we learn and preserve history. But there's a story to be learned not just in what's depicted in the tapestry, but also in the composition of the threads, in the dyes, in the weaving patterns. By understanding something about how a tapestry is made, we can learn something about when that tapestry was woven. Behind me on my slide is a tapestry of stone. It's also called a rock, and it's the way that the earth tells its stories. Just like a tapestry woven of thread, there is a story woven into this stone, a story that is unique and written by the environment in which this rock formed. Now, interestingly, environments that weave these kinds of stories are relatively rare. This is a picture of an ancient sponge skeleton that has been preserved or fossilized by a brown mineral. And by looking at the elements that are in that brown mineral, we've determined that it's the same phosphorus-rich mineral that composes our teeth. It's apatite. Now, it's already very rare for an organism to become fossilized in the geologic record, but this mechanism of fossilization is even rarer. So why was this fossil preserved for 500 million years by this rare mechanism? And how do we interpret or read a tapestry whose very presence is an indication of something unusual? Our hypothesis is that this sponge was preserved due to a symbiotic relationship between the sponge and a microorganism that releases a lot of phosphorus into its surroundings. Without this extra source of phosphorus, it would otherwise be difficult to get enough phosphorus into the sponge skeleton to make apatite. To test this hypothesis would require unraveling the tapestry at the finest level, the atomic level. Simply by living their lives, organisms can influence the atoms that go into the minerals that are forming around them in ways that are unique and even predictable to specific groups of organisms. If we look at the atoms that are in the ancient apatite, we can compare them to the atoms that are in apatite forming in our modern oceans, apatite that we know is influenced by the presence of these phosphorus-releasing microbes. If they're similar, it could suggest that the presence of the apatite in this ancient sponge is linked to the coexistence of the microbe within the sponge, potentially suggesting a codependence between the two during their lives. Now, this is a tapestry worth unraveling. We are taught an evolutionary narrative that emphasizes competition and survival of the fittest, and it can be easier to read predator-prey relationships in the geologic record, like a borehole in a snail shell or teeth marks on a bone. But does that capture the whole story, or is it merely a reflection of a limitation in our ability to read tapestries of stone? The fact that this skeleton has been preserved for 500 million years may hint at a more subtle narrative, a line in the storybook of Earth that instead of suggesting competition says, "We relied on each other," a line that is only readable by unraveling tapestries of stone. Thank you.
It makes you want to look on the ground, doesn't it? I love this, that, is anyone in here a gardener? Does anyone like to garden? Love it, love it. There's a chance for you if you want to volunteer. We're installing all new herbs in the front garden of the Stamp. So if you want to garden and you don't have a place to garden, you come see me. We are very lucky to have someone from Plant Sciences as our next person. Anshul Rana is going to talk to us a little bit about weeds as hidden reservoirs, thinking and rethinking strawberry disease management. Can't wait. Thanks so much.
Imagine a farmer walking through their strawberry fields expecting to see rows of lush red berries ready to harvest. Instead, the plants wilt, legends spread, and the fruit rots. The cause is a devastating fungal disease called strawberry anthracnose, which in extreme outbreaks have known to cause more than 50% yield losses. To understand this, and my study explores an understudied part of this field ecosystem that we often look, weeds in the strawberry fields. So the farmer do fungicide treatments and as well as do fresh planting material, but then this disease keeps on coming back. These reservoirs, or we call the silent reservoirs, harbor this pathogen, wait for the correct moment to strike back. Wait, the story doesn't finish here. It becomes more intriguing when we see how these weeds are managed in the fields. To kill these weeds, farmers spray herbicides. But interestingly, these herbicides may increase the fungal spore levels in the field, and hence these weeds can act as silent reservoirs or even more pathogenic. Now, think of these fungal spores as a fuel for the disease. More number of fungal spores, that means more living inoculum in the field, and hence higher infection risk for our beloved strawberry. This creates what we call a herbicide trap. Sadly and ironically, a management practice which was meant for a better crop production may unintentionally favors disease risk. To understand this, we study strawberry fields across season. By isolating this fungus from both strawberries and weeds, we found that some of the common weed species harbored these pathogens between strawberry growing seasons. Along with that, the whole genome sequencing data revealed they shared common ancestry and hence validating their roles as reservoirs. Furthermore, we were also interested to see how herbicides may impact the level of fungal spores, and we found that among other herbicides, one of the common used herbicides, paraquat, increased the fungal spore levels in the weeds after several days of treatment. Shocking, right? These results reveal hidden dynamics that may reshape the role of weeds in the anthracnose disease cycle. Therefore, by studying these interactions, my research will continue to study these interactions and helps farmers anticipate and mitigate these unseen disease risks, transforming strawberry disease management from just a mere spray and pray game to a smarter, strategic, and sustainable decisions in the field. Thank you.
I learned a lot. And didn't it make you want to have a strawberry? Like it was like, oh, I'll get him before that fungus spore gets it. Now, fungus spore will be your new bad word, right? When you see someone that's really making you angry, you'll be like, what a fungus spore, right? That'll be the new bad word of the day. Jess Abramson comes to us by way of psychology. And the topic of their study is hateful hometowns, area-level oppression and alcohol use among Black and white LGBTQ teens. It's great to have you here, Jess.
Have you ever wondered how your life might have been different if you'd grown up in a different hometown? Teens in the US grow up having totally different experiences. In big cities or tiny towns, in the mountains or the desert. When you're a teen, you probably don't have a lot of say in where you live. So what happens if you're growing up somewhere that harbors a lot of hate for an element of who you are? One thing that can happen is that your health can be profoundly impacted. In my field, we talk about oppression-based stress, which is the chronic additional stress people with marginalized identities face because they're targeted by prejudice and discrimination. That extra stress takes a toll on people's physical and mental health. For example, lesbian, gay, bisexual, transgender, and queer people are more likely to have an alcohol use disorder than people who aren't LGBTQ. And differences in alcohol use start as early as the teenage years. I wanted to understand why. How does the level of homophobia and transphobia in LGBT teens, home counties, and states relate to their alcohol use? Compiling many different forms of data, my research team measured the level of homophobia and transphobia in US counties and states. Then we looked at how those levels related to LGBTQ teen alcohol use in models accounting for area-level rates of teen alcohol use. We also measured levels of racism, but that story turned out to be more complicated. We found that the relationship between level of homophobia and transphobia and likelihood of recent alcohol use for LGBTQ teens was different for teens of different racial identities. Higher levels of homophobia and transphobia were associated with a lower likelihood of recent alcohol use for white LGBTQ teens and a higher likelihood of recent alcohol use for Black LGBTQ teens. Why do we see this difference? We have some preliminary results that may offer an explanation. Because they're also targeted by racism, Black LGBTQ teens face higher levels of oppression-based stress on average. When high levels of homophobia and transphobia converge with racism, Black LGBTQ teens may be using alcohol to cope. At low levels of homophobia and transphobia, white LGBTQ teens may face little oppression-based stress overall. Accepted by their peers, they may be drinking socially. This social drinking by white LGBTQ teens may be happening less in areas with higher levels of homophobia and transphobia. Understanding the effects of racism, homophobia, and transphobia is one critical step towards dismantling them and bringing us closer to a world where every teen has a chance to grow up healthy, no matter where they live.
Thank you so much, Jess. It does make you think about the places you grew up in. And thank you so much, Jess, for your science and your research. Our next presenter is Razik Zaman, and Razik is in civil and environmental engineering. And today's topic by Razik is synthetic data is better for your engine. This sounds interesting.
All right, just testing. Okay. Do you like sitting in traffic? Do you like sprinting to a bus stop because you know the next one's not coming for an hour? Do you like living way too far away from your favorite destinations? I'm willing to bet everyone's answer here is no. You don't like those inconveniences. Well, those inconveniences are the result of infrastructure decisions. The decision of where to expand roads affects traffic congestion. The decision of where to place bus stops affects transit reliability. The decision of where to build housing affects what destinations you can reach. In order to make these decisions effectively, planners rely on data from travel surveys. A travel survey, for those unfamiliar, is a questionnaire asking participants about the origins, destinations, travel times, and modes of every trip they took on a particular day. But even the most exhaustive and extensive travel surveys might hope to capture about 1 in 50,000 of the trips that happen in a year in a given region. So how do we capture information about the other 49,999 trips? Well, just as the rules of Sudoku enable us to transform a grid of 21 out of 81 numbers to a grid of a complete 81 numbers, there are analogous rules that could enable us to transform information about 1 out of 50,000 trips to information about a complete 50,000 trips. But the rules of origins, destinations, travel times, and modes is messier and far more numerous than the rules of Sudoku. In my research, I configure neural networks, which are the mathematical building blocks of modern AI, to learn and apply those messy and numerous rules. These neural networks enable me to take an input data set of 1 in 50,000 trips and produce an output data set of the complete 50,000 trips. And in my research, I like to call that kind of output data set synthetic trip data. In one experiment, we compared using my synthetic trip data against non-synthetic trip data as an input in a travel simulation. And we found that the synthetic version of the simulation matched real-life traffic counts better than the non-synthetic version. That goes to show you, if data is like oil powering the engine of scientific models, then synthetic data is like synthetic oil. It's better for your engine. And there are many engines that can benefit, whether it be traffic safety models, congestion pricing models, electric vehicle charger models, or even emergency vehicle routing models, just to name a few. Synthetic trip data leads to better models, and better models leads to better planning, and better planning leads to less time we spend sitting in traffic, sprinting to bus stops, or wishing we were closer to the places we want to go. Thank you.
All right, who needs to get their oil changed? Anyone? Have you been putting it off? And now you've just heard from Razik and you're thinking, oh my God, I need synthetic oil. Thank you so much. All right, I've learned a really important principle. In presenting our next presenter, I must remember about C in the Celtic language. What sound does it make? Anyone? Nicely done. See? Ciaran Stone, did I do it? Woo! Is a student in hearing and speech sciences. And today you're going to hear from this presenter a hopeful sip from the fountain of youth. And I will be listening a lot.
Good afternoon. Thank you all for coming. Losing some of our hearing is a thing that is likely to affect almost each and every one of us here today. Some of us to greater extents than others. But it especially affects us as we get older. Hearing loss diminishes quality of life. You can't listen to music like before, you can't talk to people like before, and this can cause social isolation, which can also lead to cognitive decline and even dementia. The WHO has identified hearing loss as the 4th leading cause of disability worldwide. My thesis is a project that asks the question, can you practice hearing in difficult conditions, basically doing strength training for your brain, in order to help older adults hear as if they're young again? The slide behind me shows how older and younger adults did when attempting to comprehend very fast speech, before and after 6 weeks of training. The training could either involve listening to speech in noisy conditions, or else it would ask people to listen to very fast, speeded up speech. You can see that if you're measuring people's ability to listen to fast speech, they do a lot better if they've had some training listening to very fast speech. That matters because it indicates that you can target specific hearing abilities. Tantalizingly, if you look at the fast speech training group, the older adults, after training, got up to the same comprehension scores as the younger adults prior to training. The magenta band indicates the pre-training scores for the younger adults. These older adults are performing as if they were young again. My thesis is doing a data analysis on the brainwave changes of these same people here. Any change to our behavior is due to a change in our brain, the natural process that we call neuroplasticity. I hypothesize that because of neuroplasticity and targeted training, the older adults' brainwaves will become more similar to the younger adults' brainwaves, with especial similarity happening for the fast speech training group. Since hearing loss is a thing that is likely to affect pretty much all of us, or at least someone we know, developing auditory cognitive training that's effective would be an inexpensive way to avoid pharmaceutical interventions. It would benefit a lot of lives greatly for a long time to come, including our loved ones, and eventually, ourselves. Thank you.
Ciaran, you just gave me a legitimate excuse. How many of you had to do the data privacy training that took like an hour, and you sped it up, right? You went to 1.5 or 2 times? Exactly. I was doing that for my hearing. That is right. I'm recalibrating my brain and finishing in time. Thank you. I was not alone in that double time. He changes his shirt 5 times. I love that about it. Anyhow, we come to our last in the first half, the pre-candidacy students that have been with us, teaching us a whole lot. Our last student is Ruiting Huang. And this student is in agriculture. And Ruiting is going to talk to us about how would you like your heirloom tomato? Now that makes you want a tomato, doesn't it? Mm-hmm. Thank you so much.
Thank you. So we all eat tomatoes, right? Okay, great. Have you ever noticed that the tomatoes in the local farmer markets often taste a little bit different with the tomatoes in the grocery store? Well, let me tell you something. Actually, those tomatoes are heirloom tomato varieties, which are so famous for their rich flavor, bright colors, and diverse shapes. But if they taste better, why don't we see them everywhere in the supermarkets? That's where my research comes from. Well, heirloom tomatoes are a little bit dramatic. They grow too tall and very easy to flop over. And also they produce fewer flowers, so which means fewer fruits. And also they get sick so easily. So, for larger scale farming, that sounds like more labor, more cost, more risk. And modern agriculture doesn't like drama. It likes predictability and also consistency. So our growers always face a choice: flavor or efficiency. One solution is the traditional breeding. For example, we cross a very tall heirloom tomato with a short variety. In their case, we're kind of between the parents is the median height. You think the problem solved? Not exactly. The traditional breeding is a bit like you shuffling a deck of cards and you mix everything together. You never know what exactly you will get. So is there any better way to do this? Absolutely. I work in the tomato lab. And yes, so we use a powerful tool called CRISPR-Cas9 system, which allows us to make precise change in the tomato genome. So it is based on a natural system which can be adapted to adjust the multiple genes in the tomato genome. So instead of crossing plants again and again for years, we can directly use CRISPR-Cas9 system to adjust the multiple genes at once in tomato genome. So to shape our heirlooms into what we're looking for. And right now we have already identified several genes related to the traits and also developed some new heirloom lines that are more flower earlier, more short in height, and also more healthy. So in the future, well, maybe in 3 years, when you walk into a grocery store, you might pick up an heirloom tomato that comes from my lab. And also, that is a real heirloom tomato that still tastes very good, very amazing for our consumers, but also can be growing at large scale efficiently for our growers in the city of Maryland. Thank you.
Now I want to show up at your lab with some mozzarella cheese and just taste everything. All right, it's halftime. There's no marching band, but there is ice cream. Now, but I need to tell you something. You need to move efficiently because we start back here in exactly 10 minutes. So get some ice cream. Okay, who feels smarter than they did coming into the room? Yes. Who's got a topic of conversation you want to tell a friend about before the day is out? Thank you all in our first group for your extraordinary sort of scholarship, but also how exactly you entertained us with intellectual thought and data and possibilities. And now we want tomatoes and strawberries. We want to hear better. We want to think about the stories we've been told. Like there's so many things that are rattling in our brains. The second half of this is people that are already at post-candidacy. So they've slogged through that early part of the journey and now they're at that precipice of, oh my God, right? Like that this sort of really, whatever the final stages that seem to go on forever, but are beautiful and will change the world. So you're going to get a chance to meet additional people that have done exceptional research and have this wonderful way of conveying their information in this second half. And we are so grateful to them for coming and joining us. So Eeshita Ghosh is this amazing person that's in cell biology and out of the College of CMNS. From ER to the edge, the pickle that could. Play that out.
So as a child, I was fascinated by touch me not plant. You touch it and it leaves closes instantly. But how does a plant know it's been touched? It has no brains, no neurons, no nervous system, yet it senses and responds with remarkable precision. The answer lies in a hidden signal, the calcium. Just like our neurons, plant uses rapid changes in calcium to send messages. When a plant experiences drought, salt stress, or even a touch, calcium rises and falls in specific patterns. These calcium signatures then act like signal telling the plant how to respond and survive. For this to work, calcium cannot move at random. It has to be tightly regulated. But how? Calcium enters the cell through ion channels. Think of these ion channels as tiny doors embedded at the outer edge of the cell, called the plasma membrane, regulating the flow of calcium. But how are these doors installed at the right position to work properly? That's where my research begins. After their production at the cell's protein factory, called the endoplasmic reticulum, or the ER, these ion channels need to travel from the ER to the edge, for which they need help. There enters the pickle. Cornichon proteins, named after the small French pickles, act as delivery coordinators, escorting these ion channels to their correct destination. But we know surprisingly very little about them. Are they just delivery proteins? Or do they play a larger role in regulating calcium balance and plant development? To answer this, I did something very simple. I deleted them from the system. Because sometimes we know the importance of something when it's gone. Without cornichons, calcium levels dropped. Roots were shorter. And the plant's internal communication system was compromised. This is critical. Roots absorb nutrient, support growth, and determine crop productivity. When calcium signaling fails, the whole plant suffers, affecting its structural integrity and health. So the pickle we thought was just a delivery option, turns out helps the plant perceive the world by maintaining calcium balance and regulating plant development. So the next time you touch a touch-me-not plant, don't just see the leaves closing, but imagine the calcium waves and the messages delivered by the pickle that could. Thank you.
Thank you, Eeshita. Now, not only do we want to have tomatoes and strawberries, but we want to have pickles. We've got this whole menu in our mind. What I love is with the microphone exploding slightly, not really. Chris Hagwood is like this figure in our world that is A.B. in the stamp, and he just appears, which is so grateful. Sydney Overton is our next presenter, a representative of electrical and computer engineering, but also a member of graduate student government and also on the Stamp Advisory Board. How about that? The magic school bus of the gut-brain axes. Ooh, go there, Sydney.
Does anybody remember that episode of the Magic School Bus where they shrink down and travel through Arnold's gut? The human digestive system is full of messengers that are working to communicate with the brain via a complex network known as the gut-brain axis. In fact, more than 90% of your body's serotonin is produced in the gastrointestinal tract. There, it helps regulate things like gut motility and inflammation. Equally important, serotonin is responsible for sending messages all over in the body, including the brain, where serotonin helps to regulate mood and sleep. Serotonin has even been found to play a crucial role in diseases like Alzheimer's and Parkinson's. We know that serotonin has a role in the gut-brain axis because when serotonin signaling in your gut is dysregulated, there's often problems also in your brain. Unfortunately, we can't measure these signals in real time. And therefore, we don't quite have an idea of how serotonin is affecting these conditions. This is, in part, due to the lack of appropriate tools to study serotonin in the gastrointestinal tract in real time. What if your doctor could shrink down, just like in the Magic School Bus, travel through your digestive tract, and see what's happening? That's what my thesis is, except instead of a Magic School Bus, I am working on developing an ingestible device that you swallow and measure serotonin as it passes through your digestive system. Imagine the ingestible device, a pill of electronics and sensors, as a school bus. After swallowing, it travels through the digestive tract. And onboard electronics, much like a bus driver, know when to stop the device for a measurement. At the stop, an onboard mechanism takes a sensor off of our bus to perform a measurement of serotonin in gut tissue. After the stop, the mechanism brings the sensor back onto the device, and it continues through the GI tract, measuring along the way. By testing all of these components, like the sensors, the electronics, and the mechanisms of this device, I've shown that this isn't magic. In fact, one of my sensors can reliably detect physiologically relevant concentrations of serotonin over 100 times. Now, we get to take all of these components and put them into a single ingestible device. From there, we can use this as a tool to study the gut, serotonin signaling, and the gut-brain connection. This will also lead to breakthroughs in the ability to create new diagnostics and new therapeutics for some of those conditions like Alzheimer's, Parkinson's, and IBD. So, what was once a magic school bus is now a real, non-invasive tool to study the gut and its connection to the brain. Thank you.
Thank you, Sydney. I'm now imagining that, like thinking about what it's going to measure. All right, no magic school bus, just a tiny little instrument going through your gut. Fatima Zulfikar is our next presenter. Coming to us by way of the College of CMNS and Chemistry, this one is an interesting one, playing with something fun. Defects, PF fun, short and bright. How cool is that? Defects to fun. Bring it on.
What if we could send tiny molecular robots into your body to image, sense, and even treat disease? Sounds like science fiction. Well, the building blocks may already exist. Imagine rolling a thin sheet of carbon atoms into a tiny tube. It's 100,000 times thinner than your hair. That is a carbon nanotube. It absorbs light and emits near-infrared fluorescence, the kind of light that can travel deep through biological tissue, which makes it very promising in nanomedicine. But nanotubes in the body? For safe excretion from the body, they must be at least less than 40 nanometers in length. But when ultra short, they stop fluorescing. Our lab discovered that defects could fix this. Now, would you want a diamond with defects? Probably not. But did you know that defects can give beautiful colors to diamonds? The same way, defects restore fluorescence in ultra-short nanotubes, helping us create FUNs, fluorescent ultra-short nanotubes. The challenge, however, is making FUNs with precise lengths while retaining this defect fluorescence. My research tackles this in 2 steps. Step 1, I take a long nanotube, modulate the molecules around it, and carefully introduce chemical tags to place precise spaced defects. Step 2, think of a chocolate bar. The defects are like the scored lines on the chocolate bar. I introduce highly reactive radicals, more like chemical scissors. Now these radicals go and attack the defect sites preferentially and segment the nanotubes into pieces. But these radicals can sometimes act like toddlers. If you're not watching, they will eat the whole chocolate bar. So I track the optical signals during the reaction to stop exactly when it's cut but not over-etched. The end products, FUNs with defects retained in the ends. And the original defect spacing now becomes the FUN length. The best part, these defects can be tuned to change color, act as sensors, and even as handles for other molecules. So someday, maybe molecular robots will no longer be just science fiction. Over to you.
Thank you. Can you visualize all of these presentations, even with only one slide? The words, the stories make you see what you don't see. Thank you so much. Our next presenter is Flávia Batista da Silva. And Flávia is from BSOS, or Behavioral and Social Sciences, studying in the Government and Politics Department. And you have a little fan club. And the topic that we'll hear a little bit about today is when citizens undermine democracy, the politics of impeachment. Bring it on.
Thank you.
Impeachment, a rare constitutional tool designed to function as democracy's emergency brake, is on the rise. In the US, 1 president was impeached twice in just 2 years. In South Korea, 2 presidents were impeached in less than a decade. And in Latin America, 14 presidents have been impeached in the last 30 years. Many scholars argue that democracy dies from the top down, when authoritarian leaders elicit polarization, or when they dismantle institutions. But these explanations focus mostly on elites. My research brings citizens to the center of the debate and asks, can their support for impeachment also weaken democracy? In fact, when we talk about impeachment, citizens play a key role through protests, political mobilization, and sometimes by filing impeachment petitions themselves. Take Brazil, for example, where citizens can directly initiate presidential impeachment. Since 1990, nearly 400 impeachment requests have been submitted, 70% of them from ordinary citizens. So citizens are not passive observers. They are active participants in deciding whether presidents stay or go. My dissertation then asks, when do citizens support impeachment and why? To answer that, I conducted survey experiments in Brazil and Colombia with about 5,000 participants. I presented them with realistic scenarios in which a president was accused of misconduct. And I varied 3 things: the president's party, the strength of the evidence, and the seriousness of the offense. Then I asked them a very simple question: should this president be removed from office? The results were striking. The strongest predictor for support for impeachment was not the seriousness of the offense. It was partisanship. When the accused president belonged to the opposing party, support for impeachment increased dramatically, even when my evidence was weak. These led me to develop the concept of instrumental impeachment. When people support removing a president, not because they broke the law, but because they disagree with them politically. And being informed about political processes did not solve the problem. In fact, knowledgeable citizens were often more consistent in defending their own side and punishing the other. When I analyzed hundreds of citizen impeachment requests from Brazil, I found 3 motivations behind them. Some citizens acted out of civic duty. Others were driven by partisanship. And some used impeachment to gain political visibility. My research shows democracy can also die from below. When citizens treat impeachment as a political weapon rather than a legal safeguard, they lower the threshold for removing elected leaders. Democracy depends not only on institutions, but on how us citizens choose to use them. And when accountability becomes partisan, even constitutional tools like impeachment can contribute to democratic erosion. Thank you so much.
Thank you. Thank you so much, Flavia.
Thank you.
Our next presenter, Adira Colton, is a student in engineering, in mechanical engineering. So it won't surprise you that Adira is going to talk to you about improving biomedical research through 3D printed glass microneedles. They can go with that little instrument that's going with your gut, and we'll have it all. Bring it on here.
So many major breakthroughs in biomedical research, from understanding how cancer spreads to gene editing with CRISPR, depend on something that most people have never heard of: a glass microneedle. Microneedles, as the name suggests, are incredibly small needles with diameters as low as 15 microns. That's almost 1/10 the size of the average human hair. Whenever a researcher needs to inject a small amount of liquid into a small biological target, they make these needles by hand in lab, leading to inconsistency from human error. And that's not the only problem. These needles clog easily, either from debris in the delivery liquid or from stuff that gets stuck in the tip during insertion. If these needles can be unclogged, it's both costly and wastes time. My lab developed a new shape for microneedles to solve this clogging issue, one with a solid point and holes on the side, which is both sharper and prevents clogging phenomena. But with this new complex shape, it's really hard to manufacture with normal methods. And so we turned to 3D printing, specifically a type of microprinting called direct laser writing, which uses a high-powered laser to solidify material point by point, allowing us to make structures as small as a grain of sand. Direct laser writing is normally done with a class of somewhat flexible materials called polymers, which include plastics and rubbers. But microneedles in labs today are made of glass, which is much stiffer. My research asks a simple question: can I 3D print these anti-clogging microneedles out of glass? The answer was no until 2023, when researchers developed a new method for glass 3D microprinting, primarily composed of polyhedral oligomeric silsesquioxane, that's 600 points in Scrabble, or POS for short. This unique molecule can be mixed with the components of a standard photoresin and printed in the same way. But once the print is done, I remove it and I put it into the oven and I bake it at 650 Celsius, that's 1200 degrees Fahrenheit. The organic components burn off and it leaves behind a pure fused silica glass microneedle. Once I make the needle, I have to test it. And I chose to do this in zebrafish embryos due to their common use in biomedical research. My needles were able to successfully puncture the stiff outer membrane of the embryo, called the chorion membrane, and we were able to successfully deliver gene editing CRISPR to over 200 embryos with the same needle, which is how it's done in labs today. Even more interestingly, the membrane of the embryo moved less when my needle punctured it compared to the standard needle, which could theoretically improve survival rates, a hypothesis that we are currently investigating. With recent developments in 3D printed glass, not only can we reinvent the glass microneedle, but any glass microtool can be redesigned into shapes that were previously impossible, allowing us to have better and faster research through adaptable biomedical tools. Thank you.
Okay, you know that you can 3D print in the library, but I do not think you can 3D print glass in the library. That's amazing. Woo. You guys are going to really change the world, I do believe. Jeet Shah is going to be our next presenter, coming to us by way of physics in the College of Math and Natural Science and Computers. The topic is, don't be a square when shape controls the quantum phase. It's great to have you here, Jeet. Thank you.
Imagine you're holding two objects made of the same material. One shaped like a square, the other like a diamond. If I zoom in the interior of these two pieces far from their edges, they look identical. The atoms in the bulk do not care about the boundary shape. This simple assumption is one of the foundational principles of physics. It is called the thermodynamic limit. It says that as a system becomes very large, the shape of its boundary does not matter. The bulk forgets the edges. My research shows that this simple assumption can fail spectacularly. We built simple models where tiny quantum magnets interact with their neighbors. Then we asked a very simple question. If we keep the microscopic physics the same, but change only the boundary shape, what happens? On a square-shaped sample, we found that the system behaves uniformly throughout. But when we rotated the boundaries by 45 degrees and made a diamond shape, something remarkable happens. Macroscopic regions emerge which are in different phases in the same material. Near the corners, the tiny quantum magnets freeze into an ordered pattern. In the center, they remain random and fluctuating. These are not just thin boundary layers. They are macroscopic regions occupying a finite fraction of the entire system, even as the system becomes infinitely large. The bulk does not forget the edges. It remembers it. So the same underlying interactions gives rise to completely different macroscopic interactions just based on the geometry. So why does this matter? Because it changes how we define phases of matter. If the bulk gets altered by the shape of the boundary, then the thermodynamic limit must be defined more carefully. Second, it is also an opportunity. In programmable quantum systems such as Rydberg atom arrays, which are grids of oversized, highly excited atoms, you can literally design boundary shapes. So the geometry can be used as a control knob to create spatially separated quantum phases. The edges are not just boundaries. They sculpt the quantum interior of matter. And that forces us to rethink one of the most foundational principles of physics, that of the thermodynamic limit. So if you want to unlock new and exciting quantum behaviors, don't be a square, be a diamond instead.
Don't be a square, be a diamond. That's what we're all going to try to be. It's joy to have MPs here. I don't know if you go downstairs and ever get anything from the coffee bar, but I swear your whole undergrad career and your grad career, same table, creature of habit, but great mind. Coming to us by way of counseling psychology. M is presenting today on the topic Decolonizing the Mind, Colonial Mentality, and LGBTQ and Filipino Americans.
Thank you. Yeah, a good part of this thesis was also written at that table. So, the last place the colonizer leaves is your mind. This is an expression that we have in Filipino American psychology, speaking to how the nearly 400 years of colonization of the Philippines continues to impact mental health today through something called colonial mentality. That is, the internalization of colonial beliefs that Filipino people, experiences, and culture are inferior to whiteness. Now, this includes gender and sexual diversity, as we know that colonization of the Philippines also included the often violent imposition of religious and cultural norms that stigmatized once cherished LGBTQ experiences. But how do these beliefs come to be? And how do they affect LGBTQ Filipino Americans today? To answer this, I conducted what we believe is the first longitudinal study with this population, surveying 152 LGBTQ Filipino Americans over a 6-month period. Around 2/3 of the sample had at least one parent born outside of the US, around 1/3 was trans, and around 1/4 was multi-ethnic. I examined how discrimination may impact experiences of colonial mentality, as well as the unique impacts of colonial mentality for LGBTQ Filipino Americans. I also looked at the role of resistance and empowerment against oppression, whether people are involved in everyday activities to address discrimination and bias within their communities as a potential protective factor of these risks. These variables were studied using validated survey measures, capturing participants' lived experiences and their beliefs and attitudes about their identities. This figure illustrates my key findings. Discrimination was positively associated with higher levels of colonial mentality. Colonial mentality was then in turn associated with feelings of conflict between one's LGBTQ and Filipino identities, or a sense that these identities were incompatible with one another. Finally, identity conflicts was associated with higher levels of psychological distress. Importantly, resistance, such as, say, addressing discrimination in your relationships or participating in protests, was protective of the association between discrimination and conflicts in identity, which means that when people engaged more in resistance, discrimination was no longer connected to feelings of conflict between identities. While it is said that the last place the colonizer leaves is the mind, this study speaks to how we can push that process along by actively working to decolonize research and clinical practice, pursuing justice in ways that honor unique histories of colonization and intersections of identity for LGBTQ Filipino Americans, as well as other marginalized communities with their own unique experiences of colonization. In empowering people to resist the systems that tell them that they are wrong, disordered, and not loved for who they are, we can neutralize the impacts of discrimination and help people find identity integration, build authentic connections, and thrive in their fullness. Thank you.
Thank you so much, Em. Michael Bonthron has been sitting here listening to everyone else because I've watched. And now Michael joins us, a student in mechanical engineering. Michael is going to speak today about Built to Change, designing structure to adapt to our needs.
Thanks. How many people here have ever ridden a bike without wearing a helmet because it's uncomfortable or you don't like the way the helmet feels? Or at least I imagine we saw a lot of them as we walked over to STAMP today. All too often, safety is our #1 priority up until it becomes too inconvenient or uncomfortable for us. As cyclists, we know we're supposed to wear bike helmets, but in 97% of fatal bike crashes, the rider isn't wearing one. Well, why? Well, flexibility and comfort often come at the expense of stiffness and protection. And just like a cyclist before a ride, as engineers, we have to make similar design choices balancing between the two. It's relatively easy for us to design a structure to be as stiff as possible, or to design a structure to be as flexible as possible. But when we try to balance both, we often end up making sacrifices or compromises between the two. The main way we compromise is we design a structure with some intermediate value for stiffness that we hope works for both cases, but often this just doesn't cut it. My research asks, what if a single structure could adapt its behavior to meet both demands? I designed structures that could be configured in multiple different ways. Each configuration has its own mechanical properties. By switching between these configurations, the same structure can go from stiff to flexible and back to stiff, depending on whatever the situation demands. So how do we do it? Well, I designed structures that are made out of tiny repeated mechanical elements, kind of like a Lego set made out of Lego blocks. Like Lego, I'm able to pattern and arrange these elements in billions of possible ways. Each pattern has its own mechanical properties. But unlike Lego, these elements aren't rigidly locked into place. When I want the structure to behave differently, I don't have to disassemble it and rebuild it like with Legos. I can simply reconfigure it to another state. These reconfigurations happen quickly, but take only a fraction of a second, and we can design the structure to reconfigure in response to all sorts of external stimulus. We can attach a mechanical actuator to push or pull it, or even a change in temperature or the detection of fluid forces. And don't be fooled, these reconfigurations can have a big impact. So far, we've designed structures that increase their stiffness up to 10 times using this principle. What this means is you can have a structure that in one state is compliant and flexible. It'll move with your body like a beanie hat. But in a fraction of a second, it can reconfigure into something that is rigid and stiff, like a bike helmet. No longer needing to sacrifice safety for comfort. What we hope is that these principles can be used to design structures that are comfortable and flexible when we want them to be, but rigid and protective when we need them to be. And stiffness isn't the only mechanical property we can reconfigure. The same ideas can be applied to designing an aircraft whose wings change shape based on cruising altitude to optimize fuel efficiency, or wind turbine blades that adjust to strengthen in response to wind gusts. Rather than designing a structure for just one condition or one environment, we can design structures that are built to adapt and change their properties in response to the environment. We can design structures that are built to last by, ironically, building them to change. Thank you.
Thank you, Michael. May we all be built to last by changing regularly. Our final presenter in this portion of the program and the program in its entirety is, no pressure, Hannah, bringing it home. Hannah Daley studies in the College of Math and Computer and Natural Science in Atmospheric and Oceanic Studies and Science. Today's topic for Hannah will be measuring methane from the sky. Do emissions rise with production? That's a good question.
It's been a busy week. Take a breath with me. How often do you think about the air you breathe? Not just when you're near a farm or behind a diesel truck, but the air that you can't see or smell. Well, whether you think about it or not, the gases in the atmosphere have the power to influence our air quality, health, and climate. Let me introduce you to methane. You can't see it, you can't smell it, but over 20 years, it traps 80 times as much heat as CO2. And right now, it is leaking from oil and gas operations all across the country. So for my research, I get in a small plane and I go looking for it. Reducing methane emissions is one of the most efficient ways to slow climate warming over the next 2 decades. That's why many states and countries are setting methane reduction targets. But in order to reduce these emissions, we first need to understand how much we are emitting now. Most states rely on US EPA inventories, but these inventories have a tendency to underestimate methane emissions, and by as much as a factor of 3. Why? Because these oil and gas emissions are based on engineered approximations and expected operating conditions, but they often miss undetected leaks and abnormal operating days. If we want our climate policies to work, we need accurate numbers. And when methane leaks go undetected, they go unfixed, and that can be an explosive mistake. So here's where my research comes in. At about 1,500 feet, flying large loops around oil and gas fields, I'm not sightseeing, I'm measuring pollution. We put instruments on a small research aircraft and measure methane upwind and downwind of a region. By comparing the methane that enters the region versus leaves, and combine that with wind measurements, we're able to determine the methane emission rate from the area below. And one place we've done this study is the Denver-Julesburg Basin in Colorado, where there's over 18,000 active oil and gas wells. There's also about 1 million heads of cattle, and cows emit methane too. So, in addition to measuring methane, we also measure tracer species that act like chemical fingerprints, linking ethane to oil and gas and acetic acid to cattle emission. That way, we're able to determine how much methane is specifically coming from oil and gas emissions. And what we found is really encouraging. Despite the fact that production is increasing, the amount of emissions per unit of production is declining. What that tells us is that mitigation strategies like improving technology and stricter regulations can work. It also shows the benefit of independent measurements and also that there's more work that needs to be done. If we want to control our emissions, we need to have accurate measurements of what they are. So by measuring methane from the sky, my research takes climate commitments and turns them into measurable action. Oh, shoot. Sorry. Turns them into measurable action. So next time you're taking a breath, think about the air that you're breathing and the science working to keep it clean. Thank you.
I'm in awe of the fact you get up in that plane and run around. I'm like, woo, that's a lot. One, can I ask everyone who has presented today to just rise for a minute? Because we want to thank you for being our best teachers of the day. Thank you so much. I also want us all to thank the judges in the back row there, somewhat in the dark, for their wisdom and time. I also want to give a big shout out to Robin and colleagues in the grad school who made this whole thing happen, and those at the college level who made all those earlier sort of competitions evolve with their help. So bravo. Now, just to remind you what will happen after we leave this place. One, you'll have a new friend who is sitting in front of you or behind you. But the other thing that will happen is that the judges will select 6 winners. And those 6 winners will be known to us on Friday, or reason enough to wait till the end of the week. And they'll each receive a $1,000 award, plus they'll be having their beautiful video that was created today morphed beautifully into something that you can take to the next level of competition. So you'll want to check 3MT on Friday to see who those 6 individuals are. 2 quick things. Every day people are doing research and writing and thinking and dreaming and really wondering what the hell they're doing some mornings. And yet, somehow they come to these amazing conclusions and inventions and ideas. And how lucky are we that our world is made better by that scholarship? And then, if you can tell us something really intriguing in 3 minutes, we are made better because now we're looking for it. We want something to go through our gut. We want something to make the methane go. Like all of that because you entertained the idea in a lab, in a research study, when you were a kid in a school bus that was somewhat small. Thank you all for being present. And also, I think is there still some ice cream left? I don't know, but you could, kind of slink by there and just see and take it with you. But thank you so much. Anything else we need, Robin, from these good folks other than applause one more time for our